everyone. My name is Yanpu Huang from the Center for Nanomedicine Research at National Cheng Kung University. Today, I will present my summer research project titled Optimizing Ultrashort Cell-Free DNA Analyzing Technology for Improving Diagnostic Efficacy in Breast Cancer. This project was supervised by Dr. Wei-Wen Huang and Dr. Wei-Bang Zhong, and it aims to improve ctDNA detection sensitivity in HR-positive, HER2-negative breast cancer through an optimized cfDNA, uscfDNA workflow. Research background. Breast cancer remains the most common malignancy among women worldwide. As known in the chart on the left, the HR-positive, HER2-negative subtype accounts for about 70% of all breast cancer cases. However, this subtype also has the lowest ctDNA detection rate, only around 33% in early-stage patients. Therefore, enhancing cfDNA detection sensitivity in this group is both clinically important and technically challenging. Recent studies from UCLA and NCKU reveal that beside the conventional 166 BP nucleosomal cfDNA, there exists a distinct population of ultrashort cfDNA, about 30 to 100 nucleotides in length. These fragments are mostly single-stranded and cannot be detected by traditional double-stranded library construction, but appear only under single-stranded library preparation. When comparing extraction methods, cryo-EM and sprite-based kits demonstrate higher recovery of these ultrashort fragments. Thus, optimizing extraction and library methods is crucial for accurately analyzing uscfDNA. Method overview. To address this challenge, our study focused on three main goals. First, to improve ctDNA detection in HR-positive, HER2-negative breast cancer. Second, to establish a uscfDNA extraction workflow using the MCF771. And third, to standardize cfDNA pre-analytical processing for blood samples. These steps together led to a short fragment-friendly cfDNA uscfDNA workflow that can be applied to multiple breast cancer subtypes and future clinical testing. DNA extraction and validation. Cellular DNA was extracted using the QIMP DNA mini-kits. All samples showed excellent purity. This validated that our workflow is stable, reproducible, and suitable for short fragment cfDNA recovery. We used the MCF771, representing the HR-positive, HER2-negative subtype, as our experimental model. Cells were treated with alpelisib, a PI3K alpha inhibitor, and trovidin, an antibody-drug conjugate. These treatments mimic different apoptosis mechanisms, allowing us to study how cfDNA and uscfDNA release changes under various stress conditions. In addition to cell-based experiments, we established a blood-based pre-analytical workflow. Blood was collected in EDTA tubes, which prevent coagulation and avoid genomic DNA contamination. Through plasma separation and density gradient centrifugation, we successfully isolated PBMC and PNN, ensuring cfDNA purity. This standardized workflow serves as a foundation for accurate plasma cfDNA analysis. This slide shows the day zero process, about 2 hours after blood collection. Freshly isolated PBMC and PNN were obtained immediately after centrifugation. This ensured that cfDNA in plasma remained intact and uncontaminated by genomic DNA leakage. By completing plasma separation within 2 hours, we preserved cfDNA purity for downstream analysis. On day 1, the culture PBMC and PNN remained morphologically intact, indicating successful separation and viable cell populations. This demonstrates that our pre-analytical workflow minimizes cellular rupture and DNA contamination. Together, the day 0 and day 1 experiments validate the standardized cfDNA preparation process for future clinical applications. Future work. In the next stage, we plan to apply this workflow to HER2-positive and triple-negative breast cancer cell models. We will integrate fragmentomics, mutation detection, and methylation analysis to establish a multi-model cfDNA analytical platform for early diagnosis and treatment monitoring. This research lays the foundation for enhancing liquid biopsy sensitivity in diverse breast cancer subtypes. Acknowledgement. I would like to express my sincere gratitude to Dr. Wei-Wen Huang and Dr. Wei-Bang Zhong for their invaluable guidance throughout this project. Special thanks to the senior members of our lab for their assistance, and to the College of Medicine, National Cheng Kung University for research funding and support. Thank you very much for listening.